Hidden genetic clues for autism are finally coming to light, and it's all thanks to a game-changing technology. For years, scientists have suspected that our old methods of reading DNA might overlook crucial information. Now, as new research emerges, that suspicion is being confirmed. But here's where it gets controversial. The very backbone of genetic research, short-read sequencing, may not be enough to fully understand autism's roots. Two groundbreaking studies, recently posted as preprints on medRxiv, showcase that long-read DNA sequencing, an advanced method that reads much longer stretches of DNA than standard approaches, can uncover autism-associated genetic variants that were previously invisible. And this is the part most people miss. These discoveries are shaking up what we thought we knew about the genetic landscape of autism. Let's break it down. Traditional short-read sequencing chops up DNA into fragments about 150 base pairs long. Scientists then use powerful computers to piece these segments back together, but complex and repetitive regions often become a muddled puzzle. Enter long-read sequencing, which captures continuous DNA fragments thousands of base pairs long. While this method comes with a heavier price tag, it provides a sharper view of complicated mutations, such as structural variants, SVs, and repeated DNA sequences, that short-read tools might gloss over. Hundreds of genes have already been linked to autism through large-scale genomic studies. Still, a huge portion of autism's genetic heritability remains unaccounted for. Why? According to Jonathan Sabat, a professor at the University of California, San Diego, our conventional sequencing strategies may be missing key structural changes in the genome. With long-read sequencing, we're finding entirely new types of mutations, he says. These are changes that short-read methods simply cannot resolve. Sabat and his team used long-read technology to assemble the complete genomes of 243 individuals from 63 autism-affected families. By comparing these results with those obtained using short-read sequencing in the same people, they discovered 15 rare, newly arisen de novo structural variants in gene-coding regions associated with autism. Notably, 3 of these would have slipped under the radar without the long-read approach. The other study took a different path, constructing near-complete, custom-made genome assemblies for 189 people from 51 families, all with at least one child whose autism had no previously identified genetic cause. Using this painstaking approach, they uncovered 3 structural variants affecting well-established autism genes, plus 9 others with likely functional impact, almost all of which were missed by short-read sequencing. Curiously, both studies found that using long-read sequencing boosted the number of identified autism-associated variants by about 4 to 6%. Not a staggering figure on face value, but Evan Eichler, a genome sciences professor involved in one of the studies, points out that if these rare genetic changes work together to raise autism risk, finding every last one becomes critical. Long-read sequencing, he says, is the only method that can get us close to that goal. Sabat's team cataloged an astonishing 44,647 structural variants not involving tandem repeats with long reads, an impressive number, especially given that 16,488 of these had escaped detection by standard short-read analysis. However, there's a twist. About 7,000 variants appeared only in the short-read data, proving that each technology has its blind spots. Is it possible, then, that a combined approach is the real answer? A majority of the detected structural changes resided outside coding regions, the so-called junk DNA. Yet the 15 de novo variants, mutations not seen in the parents and strongly linked to a child's condition, were found within crucial gene-coding exome regions. In an unexpected outcome, 3 of these were revealed solely through long-read sequencing, while others appeared only with short reads, or with both. Why does this matter? As Dr. Michael Gandal, a University of Pennsylvania professor, explains, it challenges the conventional wisdom. Short reads were long assumed to be precise enough for the tiny percentage of the genome that codes for our proteins. Clearly, that isn't always true. Here's an example. One identified variant was an in-frame duplication in the STK33 gene, leading to a dramatic expansion in the protein's structural domain, a change almost certain to alter its function. Two other variants, one new and one inherited, revealed intricate reshuffling of DNA, likely influencing gene behavior. But the opportunities don't stop there. Long-read sequencing also allowed researchers to perform phasing, piecing together which genetic variants came from each parent, and to analyze DNA methylation, an important modification associated with genetic regulation. When studying X chromosomes in girls, they found that the length of a repeated segment in the FMR1 gene matched with methylation levels, but wasn't linked to X chromosome inactivation. This finding echoes earlier research, suggesting consistency but raising new questions about how these patterns influence autism. Meanwhile, the second research team, led by Eichle, compared their participants' genomes to a comprehensive human, pangenome, and large datasets of known structural variations. This strategy trimmed an initial catalog of over 33,000 variants down to just 46 especially intriguing differences in 87 children. Among these, 3 de novo variants, 2 missed by short reads, overlapped known autism genes. An additional 9 turned up in regulatory or coding areas, with 7 escaping detection by old methods. Controversially, they also found something unusual. 6 individuals each carried a rare autism-linked variant not just once, but twice on both copies of the gene. With such homozygous cases statistically rare, this was a surprise to experts like Dr. Charleston Chiang at USC, who wondered aloud about how these combinations arise. Interestingly, these double variant cases were not seen in the Sabat study. Is this simply due to different study designs or populations, or does it speak to deeper, undiscovered trends in autism genetics? Yet, there's a catch. Both studies acknowledge the elephant in the room. The current cost of long-read sequencing limits how many people can be studied. But as technology advances and costs drop, expect this method to revolutionize genetic research far beyond autism. As Gandel concludes, this is just the tip of the iceberg. So what do you think? Is long-read sequencing the silver bullet for uncovering autism's genetic roots, or should researchers stick to a combination of methods? Could what we've long considered good enough in genetic science actually be holding us back from major breakthroughs? Sound off with your thoughts, disagreements, or bold predictions in the comments below. The future of autism genetics may very well depend on it.